This is our lab report number 2. It will be based on staining and it will include different staining techniques such as simple, gram, acid fast, capsule, and endospore stain. This will be presented by Massiel Martinez, Juan Osiel Sanchez, and Mauricio Sanchez. Beginning with the introduction. The objective of this lab is to learn how to properly use a compound microscope, how to prepare bacterial smears from broth and agar cultures, as well as to gain an understanding of how to perform these different staining techniques in order to detect and identify the type of bacteria based on their cell characteristics. For example, for the gram stain, we will be able to determine whether it is a gram positive or a gram negative. We will be performing aseptic techniques throughout the entire experiment to protect ourselves as we will be dealing with a numerous amount of bacteria. In this slide, it demonstrates the materials and methods used for this experiment. As you can see, there are 5 different cultures used, such as Staphylococcus aureus, Bacillus cereus, Salmonella typhimurium, Klebsiella pneumoniae, and Escherichia coli. The 2 types of media used in this experiment would include blood agar and tryptic soy broth. As far as stains go, there are many stains used throughout the experiment, such as crystal violet, safranin, quinone, and a few others. Other materials that were used during this experiment would include an inoculating loop, a compound microscope, glass slides, Bunsen burner, and a few other materials that, as you can see, are listed here on the slide. All of these materials were used to perform different staining techniques, such as a gram stain, acid fast stain, capsule stain, endospore, and a simple stain. Before we begin with the staining procedure, we would first need to do a smear preparation, one from a solid medium and another from a liquid medium. The steps are very similar, however, they are different. For example, for a solid culture, we would first obtain a clean glass slide, make a big circle using a wax pencil. Then, with the inoculating loop, we would transfer a drop of water onto the circle, proceed by sterilizing the loop, grabbing a colony, and spraying the bacteria in the water to fill in the circle on the slide. Then, we will place the glass slide to air dry on a warming block. Once it is dried, the slide will need to be heat fixed and it is ready for the staining procedure. As for the liquid culture, water would not be involved since it is already liquid. We would obtain a clean glass slide, make a big circle with a wax pencil. Then we would gently swirl the tube containing the bacteria. Then we would proceed by sterilizing the inoculating loop and transferring a drop of the culture broth to the slide and spreading it to fill in the circle. Then we will place the glass slide to air dry on a warming block, and only for the gram stain would we need to heat fix the slide. An image has been included to demonstrate a more simple detailed visual on the types of smear preparation. As you can see, on the left side is the step for the solid medium, on the right side is the step for the liquid medium, but they both end by air drying the slide. And on the bottom, it demonstrates that it will be heat fixed, which in this case for the liquid culture, it would only be for the gram staining.
For the simple stain, we did a smear preparation and heat fix for the S. aureus bacteria, and we flooded this slide with crystal violet and let that sit for a minute. And then after the minute passed, we rinsed with water and dried it with the bibulus paper, and then it was ready to view under the microscope. And for the gram stain, we did the smear and heat fixing for S. aureus, B. cereus, S. typhimurium, and the mixed, which was S. aureus and E. coli, and the 2 unknowns, unknown 201 and 102, both the broth and the culture. And for these slides, we flooded them with crystal violet stain, let that sit for a minute, then rinsed with water for 5 seconds, and then flooded with the iodine stain and let that sit for 2 minutes. Then we rinsed with water, decolorized the slides with acetone, and we immediately flushed with water after we put the acetone. And then finally, we flooded the slides with safranin stain and let it sit for a minute, rinsed with water, and then dried it with the bibulus paper and viewed them under the microscope. And on the right, you can see what the gram positive and gram negative looks like for the gram stain. Gram positive it appears purple and gram negative appears pink. For the acid fast stain, we used a pre-prepared slide of Mycobacterium sp. We flooded this slide with the carbofusion stain and let it sit for 2 minutes. Then we rinsed it with water and used the acid alcohol decolorizer to remove the color from the slide. Then we rinsed it with water, shook off the excess moisture, and stained with methylene blue. We let that sit for 30 seconds, and then afterwards we rinsed with water and dried it with the bibulus paper, and then it was ready to view under the microscope. For the capsule stain, we added a small drop of crystal violet to the slide, and then we aseptically added a loop full of K pneumoniae to the slide. We made sure to heat the loop before we added it, and we mixed it with a dye, and then we used another clean slide to drag the liquid over the slide into like a thin film, and we let that sit for 5 to 7 minutes until it dried, and then it was ready to view under the microscope. And on the right you can see towards the top it's the acid fast stain, and towards the bottom it's the capsule stain.
For the endospore stain, we would begin by preparing a heat-fixed smear of Bacillus subtilis. Then we would place the slide on the staining rack. We would cover the smear with filter paper and then saturate the filter paper with malachite green. Then we would heat the slide over a Bunsen burner until steam appears. Once the steam appears, we would set it aside until it disappears, and we would repeat this for a total time of about 5 minutes. We must try to avoid the slide from drying out and avoid excess flooding. Then we would remove the slide, let it cool down, and then rinse with water for 30 seconds. Then we would rinse with water for 5 seconds, counterstain with safranin for 1 minute, rinse with water for 5 seconds again, and then we would blot dry with bibulous paper and then examine under oil immersion. What we observe should appear as the image provided in the slide. Endospores appear greenish in color while vegetative cells appear red. The difference between the two is that a vegetative cell is a living active cell that could reproduce, while an endospore is a spore in a capsule that is resistant to harsh conditions.
These are our observations for the stains. We don't have the pictures for each of the stains. That's on the next slides for results. But for the simple stain, we observed in the B series cells, they looked blue in color and they were rod-shaped. And in the gram stain, the S. aureus cells were purple and round-shaped. The B series cells were purple rod-shaped. The S. typhimurium had a entirely purple background and with a few pink rod-shaped cells, and then the mixed culture had pink rod-shaped cells and purple round-shaped cells, and the unknown 102 was pink and rod-shaped, and the unknown 201 was purple and rod-shaped. For the acid fast, the Mycobacterium sp was stained red and appeared rod-shaped, and in the capsule stain, K pneumoniae appeared blue, a light blue color, and rod-shaped. And in the endospore stain, the B subtilis appeared pink. There was pink and red rod-shaped cells, and also some green-blue rod-shaped cells.
This is our results for our simple stain, which we conducted on Bacillus cereus by using methylene blue. Here from these results we can see that B cereus cells will appear uniformly colored in blue, and for its morphology we observe the general morphology of Bacillus cells, which is a gram-positive bacteria, but typically appears as a rod-shaped bacilli cells. And with its arrangement here we can see they are in single cells, pairs, and in chains. Next, we performed a gram stain on several microorganisms, such as our unknown 102, unknown 201, S. aureus, and a mixed culture that contains S. aureus and E. coli, which serves or provides us as a control. And here this will contains a gram-positive cocci and a gram-negative bacilli, which allows us to verify if our staining was done correctly. On the far left, beginning with our unknown 102, After performing our gram stain results, we can see that unknown 102 is a gram negative with a bacilli shaped. Following our unknown 201, we can see that this microorganism is a gram positive bacteria, and it is also a bacilli rod shaped as well. Now, with our mixed culture, here we can see our S. aureus is our gram positive, which is those tiny little purple red specks, because S. aureus is actually gram positive, and it is cocci shaped, as our E. coli in the very background of it, we can see is a gram negative, and it is that bacilli shape. And on the far right, again, performing a gram stain just on S. aureus by itself, we can compare from our mixed culture that it matches with it, and it is a gram positive cocci. These are our results for a gram stain for B. cereus. We see that B. cereus is a gram positive bacteria, and its morphology appears as a rod-shaped bacilli cells, and its arrangement we can see that they are in single cells, pairs, and in chains. On the right is our result for the gram stain for S. typherium. Typically, S. typherium is a gram negative bacterium, which will appear pink or red under the microscope. But here on this image that we have here, it appears a semi very dark pinkish, or would I say purple, but their cell shapes are typically rod-shaped bacilli and are elongated and have a cylindrical shape. Perhaps this may happen due to, since first during the gram staining process, the crystal violet stain initially colors the cells purple, but when washed with the decolorizer of acetone, the outer membrane probably wasn't as disrupted and didn't allow the stain to completely wash away. So therefore, maybe this is why it appears a little bit more purple. Then we performed our acid fast stain on a slide of Mycobacterium species. Here, this is a positive acid fast reaction because Mycobacterium species are acid fast, which means they resist the decolorization with acid alcohol during the staining process, and as a result, the acid fast bacterium retain the primary stain, which is a carbofusion, and appear bright red or pink under the microscope, as we can see here that our bacteria is that bright pink. As for the cell shape, Mycobacterium species are typically slender or rod-shaped bacteria, meaning they are bacilli and can be straight or slightly curved. Here we performed the capsule stain on K. pneumoniae, and these were results. We can see those definitive capsules produced by K. pneumoniae, which are typically not stained by the primary or the negative stains. Instead, they appear as those clear halos, as we can see in the image, or zones surrounding the stained bacterial cells. These halos are a characteristic feature of capsules and indicate the presence of the protective polysaccharide capsule. It's important to note that these capsules are the significant virulence factor for K. pneumoniae and play a role in protecting the bacterium. And lastly, we performed an endospore stain on Bacillus subtilis, and these were results beginning with what is the pink stained um, bacteria. Those are considered the vegetative cells of Bacillus subtilis, which appear as the pink or red under the microscope. This is due to the saffron counterstain, which stains the vegetative cells but does not penetrate the endospores. Now, what is important is those endospores of the Bacillus subtilis, which appear as the bright green structures within or adjacent to the vegetative cells. On the image on the left, looking at it closely, we can see those green endospores. And more importantly, those endospores are highly resistant structures formed by certain bacterial species to survive unfavorable environmental conditions.
Our conclusions for this lab. For the simple stain, which tells us of the cell's shape, size, and arrangement, we saw that B series had large rod-shaped cells, and in some areas they were very clumped together and formed chains. Now for the gram stain, which tells us about the cell wall characteristics, and if they appear purple, that's gram positive. If they appear pink or red, it's gram negative. The gram positive means the cell walls are thick and made of peptidoglycan, and the gram negative means that they have thin cell walls. For S. aureus, it was gram positive. B. cereus was gram positive also. S. typhimurium was gram negative. The mixed culture showed gram positive and gram negative. Gram positive for the S. aureus cells and gram negative for the E. coli cells. The unknown 102 was gram negative and the unknown 201 was gram positive. For the acid fast stain, which tells us also about the cell walls, whether they're resistant to decolorization by acids. If they appear red, that means they're acid fast, and if they appear blue, that means they're non-acid fast bacteria. For the Mycobacterium sp, they appeared red, meaning they were acid-fast bacilli. For the capsule stain, this tells us if a capsule is present, if it's capsulated or non-capsulated. K. pneumoniae appeared light blue around the cell, meaning that it was capsulated. If it was non-capsulated, it would have had a dark blue color around the cell. For the endospore stain, which it helps to identify the presence of an endospore and tells us if the bacteria produce endospores, the endospores will appear green and the vegetative cells will appear red. And here we did see green and red colors, which showed the presence of endospores and the vegetative cells for B. subtilis. These were two of the resources that were important for this lab to interpret the results and to write the results. The lab manual, pages 14 through 17, and the lab journal observations that we wrote. And that concludes our video lab report 2. Thank you for watching.